everyone and welcome to Cause Effect. This is a channel that aims to spread scientific researches and innovative projects with the goal to make the knowledge accessible for everyone. My name is Ana Rita Batista, and today I have as my guest the researcher Arianna Fumagalli. Nowadays she's a postdoc in the Jarno Drost group at the Prinses Máxima Center for Pediatric Oncology in Utrecht, Netherlands. Arianna, welcome. Now you have the word to greet the people who see us.
Hello everyone. I'm really happy this morning to be here and discuss about science with you all.
So Arianna worked during her PhD in the Van Reenen group at the Polytechnic Institute in the Netherlands Cancer Institute. Here she did her research about colorectal cancer and developed new methods to study the metastases of this cancer, resulting in her PhD thesis, Cancer Cell Plasticity and Niche Independency-Derived Metastases in Colorectal Cancer. So colorectal cancer is one of the most common types of cancer worldwide and the second leading cause of deaths in the Western world. Most of the deaths related to colorectal cancer are the results of metastases in other organs. Arianna, to begin with and to simplify the information for everyone who is watching us, what are these metastases and why or how do they happen?
Yeah, so metastases are also known as secondary tumors. So basically they are derived from the spread of tumor cells from the primary sites, that in this case was the colon, the intestine, to other organs via, for example, the blood or the lymphatic vessels. And these other organs are, for example, the liver or the lungs. And in those organs, there are new tumors that arise over time. And eventually, unfortunately, they will take over the entire organ, so the organs will not function properly anymore.
Okay. Is there a predisposition for it to appearance? Is there someone that is more prone to have this type of cancer?
So for colorectal cancer, there are some genetic predispositions. They are not very common. Let's say that only 10% of all the colorectal cancer have an underlying genetic predisposition. There are some syndromes that are very famous, such as, for example, Lynch syndrome or FAP. So there are patients that are already early on during age developing polyps, which are benign tumors that later on, unfortunately, develop into malignant cancer. Although the vast majority of the colorectal cancer patients have sporadic tumors, so they are basically just derived from bad luck.
Okay. During your PhD, you established a new model for studying colorectal cancer, the metastases. What new model was this?
Yeah, so let's say that in the last 15 years, starting from colorectal cancer biology in general intestinal biology, there was a very important technological advance that was the establishment of organoids. Organoids are in vitro cultures, so cell cultures that you can maintain in dishes. But they have a particularity. They are organizing themselves into 3D structures that are representative of the organ of origin. So this particular type of cell culture can be generated by basically any organ of our body, and also from organs with disease, such as, for example, tumors. So what I use this technology to generate organoids from colorectal tumors. And in particular, I developed a model where I could visually distinguish stem cells in a particular color from the rest of the cells, so more differentiated cells in another color. And what we did then is to generate primary tumors in vivo with these organoids, so being able to follow stem cells and no stem cells, And eventually these tumors would also lead to formation of metastases in other organs, such as lung and liver. And also there we could look at the stem cells versus the other cells, and for example, understand which of these two cell types are capable of generating metastases.
Mm-hmm. And for the people that don't understand this very well, what are these stem cells?
Yeah, so stem cells are considered to be the cell of origin of cancer, and also they basically are the cells that are capable in every organ that, yeah, indeed is part of our body to regenerate and to maintain cellular homeostasis. And for cellular homeostasis, basically what I intend is that just to maintain the organ and our epithelial, so our tissues, as they are. So they are a small population of cells that are capable of generating all the other cells of our organism.
Okay. And which are the differences between this model that you find and the mainly used nowadays?
I think the main innovation is that indeed we can follow very easily with the microscopy. So by really visually looking at these cells, which cells are stem cells and which are not, and what are the interconnections between these two different cell types. So what we we're able to see is that, for example, cells that initially are not stem cells, so for example, they are of a red color, they somehow, in certain conditions, such as, for example, when they arrive to other organs, such as, for example, the liver or the lungs, they transition to stem cells again. And so they become of another color, of green color, for example, so they are stem cells, and then they can start over a new tumor where they are.
Okay, and this innovation, this new method, can be helpful in other types of cancer and not only colorectal cancer, or you don't know that yet?
No, I think it could be helpful. The issue there is more to understand how to generate cells that have that are stem cells, so how to generate a model in which stem cells can be truthfully recognized as such. So what is very difficult sometimes in many other types of tumor is to identify the stem cell population over the rest of the tumor cells.
Okay. And what would the treatment be like if we were applying it on a patient? How would it be based on this discovery?
Yeah, so in the last 10 years, actually, a lot of scientists around the world have been focused their energy on trying to develop stem cell therapies, so specific drugs that actually target cancer stem cell. Because indeed, if the stem cells are at the beginning of the tumor, if you imagine to kill these, then you should be able to eradicate, so to kill the entire tumor.
Okay.
What's happened with the study that I was part of is that we understood that actually that might not be enough, because in the absence of stem cells, other cells of the cancer are able to regenerate the stem cells. So what I think now we envision is that on top of a stem cell therapy, you would also be able to target this plasticity mechanism that actually is the one responsible of the transition from non-stem cell to stem cell again.
Mm-hmm. And now, how it is going, the investigation? It will be applied in a treatment? How are the things?
No, so I think we are still at a bit of basic science phase. So what I know is that other groups around the world have taken over the organoids that I generated with the stem cell and the non-stem cell markers. And they are trying to understand what is at the basis of this plastic transition between non-stem cell and stem cell. And that is the step one, because if you understand the biological mechanism, so which molecule is actually responsible for this transition, then you can try to design a drug to target it.
Okay, so how many years do we have to wait for the treatment?
I think at least other 5.
Okay, 5 more years. Okay. For the people that are watching us and might be concerned about having predisposition to have this type of problem, is there any way to avoid it? I don't know.
To avoid it, well, you know, I'm a biologist, so for me it's a bit difficult to answer this question, but what I would suggest is to go to your doctor and just do screenings to try to be as to, you know, be prevention and be checked out often.
If it's a genetic problem, the people can know it previously if they have this
so if it is
predisposition?
familiar, normally what happens is that you have in your family someone else that unfortunately got already sick, and they probably had a genetic test that indeed revealed that there is a genetic predisposition, so there is a specific mutation in your DNA that might be responsible for this disease, and then you can ask to have also a genetic test as a member of the family and to find out.
And what are the expectations for the future? Your expectations?
My expectation for the future, in which sense?
With this discovery.
With this discovery, okay. Yes. Well, so indeed, this discovery was pretty innovative. The concept of plasticity in metastases was not there before. I think it's in the line of, let's say, a new scientific path, you know, that is at the basis of, you know, understanding that there are, like, for example, some regenerative processes that are really common in cancer and are responsible indeed for cancer spread or metastasis formation. So my hope is that many other groups will join in forces to indeed push on science in this direction and trying to find out more of the mechanisms and why metastases happen.
Okay.
Because this is the only way to find new therapies for patients.
Okay. Now you are still working in cancer, right? But with children.
Yeah. Okay. Yeah. So I started my postdoc now 2 years ago at the Princess Máxima Center. Is a hospital for pediatric oncology. So from adult cancer, colorectal cancer, I moved to pediatric cancer research, which of course is really challenging and actually not a big field. So there is not a lot of knowledge about it. And that's why it's so interesting. And I think is also nice, you know, as a personal contribution that I can give to the science and to the patients as well.
Okay. Arianna, thank you so much for this interview. I wish you lots of success and in your researches, and that maybe we can get back talking about a new discovery.
Yeah, sure.
Now you can also say goodbye to the people who see us.
Okay, goodbye. See you soon.
Thank you so much to everyone that watched this video. I hope you enjoyed it. Please don't forget to subscribe to our channel so you can support our project, and give a thumbs up to this video if you liked it. And this was Cause Effect. See you soon.